One of the things that Watson and Crick did in their publication on DNA structure is that they also went on to propose a model for DNA replication. It's important to keep in mind that the DNA molecule has to be a molecule that can be copied quite easily and perfectly, because every time a cell is going to replicate, it's going to have to copy the DNA. So what we see on this slide is a little excerpt from their paper. They said, now our model for deoxyribonucleic acid is, in effect, a pair of templates, each of which is complementary to the other. We imagine that prior to duplication, the hydrogen bonds are broken and the two chains unwind and separate. Each chain then acts as a template for the formation onto itself of a new companion chain, so that eventually we shall have two pairs of chains where we only had one before. Moreover, the sequence of the pairs of bases will have been duplicated exactly. So, in other words, what they were saying is that we're going to start off with a double-stranded DNA molecule, as you see right here. That molecule has the bases on the inside of the helix, and those bases we see the complementary base pairs, so the A is always across from the T and the C is always across from the G. Now they said that if we're getting ready to replicate the DNA, we imagine that first off, the hydrogen bonds are going to be broken. So if the hydrogen bonds are broken, keep in mind that the hydrogen bonds are what is holding the two strands together. So if we can break those bonds, we will separate or pull apart the two strands. Then they said, if we have pulled apart these two strands, we can have them separate from each other, and we can go right down here and we ought to be able to figure out how to build the second strand. So in other words, we know that across from an A is always a T, across from a C is always a G, across from a T is an A, and so on the whole way down. And we really don't need to look at this strand over here at all to figure that out, so long as we understand the base pairing rules. So that we see right here. They said you could pull the strands apart, and now we can simply copy right on top of each one of those exactly what the other half of the molecule ought to be. And if we do this, then we end up with a replicated double-stranded molecule of DNA. So we started with one double-stranded molecule, now we've got two double-stranded molecules. And if you look at them closely, you will notice that they're exactly the same. Now in this picture, the dark blue is representing the old DNA, and the light blue is representing the new DNA. Notice here that when we have these two double-stranded DNA molecules, each one of them has one completely old strand of DNA connected to one completely new strand of DNA. That is what is called the semi-conservative model of DNA replication. The semi-conservative model of DNA replication is considered to be the accepted model today. There were a number of other molecules or other models of DNA replication that were out there, but a lot of experiments and work went into this particular model and kind of proving that this one was the true case and not the others. So if we look at this DNA sequence here, we know what the sequence of DNA is. We ought to be able to figure out what the complementary strand sequence would be. What we mean by complementary is that we should be able to figure out what the opposite strand, or the strand that is hydrogen bonded to this one, would be. So to do that, we would just need to know our base pairing rules. If we keep in mind that across from a C is a G, we can write that right next to it. Across from a T would be an A would be a T, and so on. We can then come up with the complementary strand. So what I've drawn right underneath here would be considered the complementary strand. And if we just add another layer of complexity onto that, if we say that this top strand, this is the 5' end and this is the 3' end, then we know that this bottom strand is going to be opposite that, keeping in mind that the two strands of DNA are always anti-parallel to each other. You do want to make sure that you know the base pairing rules of DNA, so that if you're given a DNA sequence, you are going to be able to come up with the complementary strand, or the sequence of the opposite strand. We also want to make sure that we understand those strands are going to be anti-parallel to each other. Now that we know kind of the general scheme of how the DNA is going to be replicated, now we're going to talk about the actual process of DNA replication. Now I do want to point out that the process we're talking about here is what is happening during S phase of interphase. So keep in mind the S phase of interphase, when we discussed it before, we simply said the DNA gets copied. And we really left it at that. We said the DNA gets copied, and that's it. Well, what was happening, if you kind of envision what we looked at in the pictures before, we started off with a chromosome that was composed of one chromatid. Then by the time we finished S phase, we had a chromosome that was composed of two chromatids. And we then called that the replicated chromosome. Well, what we were looking at was basically taking a double-stranded DNA molecule. It was wrapped around a bunch of protein, but it was one double-stranded molecule. And if we follow what we just described, we're going to separate these two strands, and then we're going to build right on top of each one of them, so that ultimately we do end up with our two double-stranded DNA molecules, but in this case they happen to be tacked together at the centromere, so we call it the replicated chromosome. So that's the process that we're going to look at here when we talk about DNA replication. Now we will have a number of enzymes involved that are going to help make sure that the DNA always gets copied correctly. And then we're also going to have some enzymes involved that are going to help make sure that if it's not copied correctly, we do get that problem fixed. So if we begin talking about all of this, we're going to have lots of DNA that has to be copied inside the cell. And because there is so much DNA that has to be copied, and it needs to be copied in a pretty timely fashion, we will have lots of starting points. So those starting points are going to be what we call origins of replication. What the origin of replication is going to be is this is the initial separation point. So we have our initial strand separation occur here. And if we're separating the strands, what we're really doing is we are breaking hydrogen bonds. Because keep in mind that the hydrogen bonds are what is really specifically holding the two strands together. So I'll try to draw these pictures as neatly as I possibly can, but it may get a little bit sloppy. So we're going to have a DNA molecule that is going to start to peel apart in specific little locations. So that's what we're seeing happen right here and right here. Now all of the rest of this would still be hydrogen bonded together. Okay, so I'll put our primes on here so we can see that this double-stranded DNA molecule is anti-parallel. So here we have actually separated the two strands of DNA. The hydrogen bonds between the bases that were holding them together have been broken. And we will have a specific enzyme that's involved in separating or pulling those strands apart from each other. The key here is that if we want to actually see the sequence of the DNA or read the sequence of the DNA, which is all in the bases, keep in mind that the bases are hidden in the middle of the helix. So to read them, we really do have to separate the two strands from each other so that we can actually look at the DNA, read the letters, and then we can start to build some more DNA right on top. We're going to have sets of enzymes that are associated with each one of these areas of the helix that we have separated from each other. Now, what we're looking at here, this would be called a replication bubble. And notice that the replication bubble is going to have two forks. We've got a fork right here and a fork right here. So by fork, we're talking about like a fork in the road. So we had a double-stranded molecule that was together, and now it's peeled apart into two different pieces. We're going to be zooming in to a replication fork at various times as we go through this process of DNA replication. So I do just want to emphasize that it is half of the replication bubble. It's not the entire bubble, and it's not all by itself there inside the cell. If we have lots of origins of replication, as we will have inside the cell, This is going to enable us to copy the DNA in a pretty quick fashion. So we're talking about hundreds, if not thousands of these on each chromosome that we have inside the cell. And keep in mind that for humans, we've got 46 chromosomes. So we're going to have at least a couple hundred origins of replication on each of those 46 chromosomes. Now, if we zoom in to that replication bubble, I'm going to draw it a little larger here. If we look at this replication bubble, the first thing that we had to do is we have to separate the strands from each other. Okay, so here we draw our double helix. We're going to have bases on the inside here, so I'll go ahead and write some letters here just to emphasize the fact that we do have letters here. They would be the whole way through, although I'm not going to write it on the whole thing. And keep in mind that these are going to be complementary to what we see on the top. Okay, so those would have been hydrogen bonding to each other before. When we have these origins of replication, we have to separate the strands. So our first enzyme, our very first enzyme in all of this, its job is to separate the strands from each other. So this enzyme's name is going to be helicase. Helicase is going to act somewhat like a little bulldozer that's going to be operating at each of the replication forks, and it is essentially running right through here and just busting open or breaking the hydrogen bonds that are holding these two strands together. Okay, so it is ripping through, kind of unzipping the DNA, you can think of it as. That's what's creating the replication fork. Now keep in mind that if these are ripping through there and separating the strands, the replication fork is definitely moving as well. And this replication bubble that we have is going to be growing or expanding. Now as we do that, we would also have bases right here where we have just opened up the DNA. Now as we separate them from each other, keep in mind that the A that we have right here is still going to want to hydrogen bond to this T. And this T up here is still going to want to hydrogen bond to the A, and so on. So they want to stick together with each other. So we do have to do something to keep the strands propped open from each other, prevent them from basically snapping right back together. So that is where our second set of molecules is going to come into play. These molecules would be what we call single-stranded binding proteins. They're called single-stranded binding proteins because they are binding to single-stranded protein or single-stranded DNA. And they're going to basically wrap themselves around the DNA right here next to the fork. Okay, so we would have some over here as well, covering up those bases that have just been peeled apart and separated from each other. The purpose of these is to keep the strands propped open. Now notice that the single-stranded binding proteins are only going to be right there at the replication forks. We're not going to take them and line the entire molecule of DNA with those. And because we're not going to do that, it would require so much protein, we're not going to just peel the two strands of DNA apart from each other completely. We're just going to open up small little sections at a time. And that's why we have a replication bubble, not just two separate individual strands floating around inside the cell. If we did completely separate them from each other, they would just snap right back together because we're not going to have the means to really keep them from sticking. So we've got our DNA opened by means of the helicase enzyme, and we've got it propped open using the single-stranded binding proteins. Once we have this accomplished, we can now see some single-stranded DNA, so we can read the nucleotides, the bases that are there, and we should be able to determine then what we want to put in the new DNA that we're going to make. Now, the enzyme that's going to be making the new DNA for us, its name is called DNA polymerase. So DNA polymerase which is often just called DNA pol, its job is to make DNA. So it's going to make DNA by reading DNA. So it's looking at the old DNA, seeing the bases that are there, and it knows the base pairing rules. So if DNA polymerase sees an A, it knows that it should put a T across from that. If it sees a C, it knows that it should put a G across from that. So DNA polymerase is going to be reading the old DNA. Now, a few things to keep in mind with DNA polymerase is that it is going to be adding nucleotides to a 3' end. It always adds to a 3' end. What that means is that it must have a 3' end that it can actually add onto. So in other words, it cannot initiate the process. So because it can't initiate the process, this is going to require us to have another enzyme that is capable of initiating. But it's going to add nucleotides to a 3' end. Notice that when it does that, what it's really doing is it's making one of those phosphodiester bonds that we talked about before. So it's making phosphodiester bonds, or linkages, if you want to call them a linkage. And it also works anti-parallel to the template. So our template would be the old DNA. And the old DNA is going to be 5' to 3', and we'll be working in the opposite direction because we've got to make it anti-parallel to the template. Okay, so we're always going to be making our new DNA anti-parallel on top of the old DNA. Now, if we go back to our replication fork that we had our replication bubble. We'll put our primes back on here. We were opening this up with our helicase. We had it propped open with our single-stranded binding proteins. And we had some letters up here on the top. So if we just draw a few of them there. Because DNA polymerase cannot initiate, we have to have that other enzyme that's going to come in and do that for us. And that enzyme's name is primase. Primase makes an RNA primer. So we call it an RNA primer because the nucleotides are a little bit different. These nucleotides would have ribose sugar instead of deoxyribose. And we would also not have the thymine bases. In its place, we would have the uracil bases. So this is going to be built right on top of the DNA. So if we look here, because it's RNA, we're going to build it right on top, but I've changed the color here to emphasize, this would be a U, to emphasize that this is RNA. So if you would normally put a T in, now we're going to put a U. And notice that we're still using our base pairing rules. So down here, we're going to have the same thing take place. And when we build the primer using our primase enzyme right on top of the DNA, it is also going to be anti-parallel to the template. What that means is if you look at the top strand, this over here was the 5' end. So here was a 5' end. That means this is going to be a 3' end. And this over here is going to be a 5' end. And we'll have the opposite thing taking place down here. The reason we have that primer placed on top of the DNA as it is because this now provides a 3' end that the DNA polymerase is going to be able to add onto. So that's exactly what the DNA polymerase is going to do. It's now going to come up here to the 3' end, and it is going to start adding nucleotides onto it. So it's going to do that by, again, following base pairing rules. It would look at whatever letters happen to be over here, and it would be putting the opposite letter or base in there. and we're going to extend the primer with some DNA on the end. Keep in mind we're hydrogen bonding this to the template. We would do the same thing down here on the bottom. Okay, we're adding on to our 3' end. Now, DNA polymerase will continue to add on to that 3' end. So this becomes the 3' end here. This becomes the 3' end here. And as it continues, the helicase enzyme is going to keep peeling the strands apart from each other, and those single-stranded binding proteins will continue to move with the fork. Now, DNA polymerase is incapable of adding on to a 5' end. So that does present us with a little bit of a problem right here. And then also right here, because we can't simply come on here and just add DNA onto the end. So instead, what must take place in that case is that we have to have that primase enzyme come in again, and that primase enzyme is going to have to make yet another primer. So the primase enzyme will back up as far as it can, and it will add a primer right on top of the template. This is again going to be anti-parallel. We will have the same thing take place down here. And this primer, as we said before, then provides a 3' end that the DNA polymerase can add onto. So DNA polymerase will come in yet again, and it's going to extend the 3' end and make DNA. So this is now becoming our 3' end here. We have the same thing happening down here. So one way to look at DNA synthesis is to say that the 3' end is the end that's growing. That's the end that's always getting longer. The 5' end stays stationary, doesn't go anywhere. Now, because we're trying to make DNA, we don't really want a little bit of RNA connected to some DNA, then another little piece of RNA, more DNA, as we see on this slide. We don't want the little blue, some red, little blue, some red. So at this point, what we need to do is we're going to remove the RNA primer and get rid of it. The RNA primer was there to provide a starting point for the DNA polymerase. We've already used it for that, so it has served its purpose and we're ready to get rid of it. So DNA polymerase is capable of actually removing that primer from the DNA. So DNA polymerase is going to remove the primer. When it comes in here and removes this primer, it's going to remove it, and at the same time, it's going to fill in right underneath it. So we're removing, we're filling in. As we do that, the 3' end is getting longer again. So this is now our 3' end. This 3' end is getting longer down here as well. And DNA polymerase will extend that 3' end until it runs into what was the 5' end of the previous strand. What it cannot do is DNA polymerase is incapable of making that last phosphodiester bond. So the one that's going to connect the last 3' end to the 5' end, and same thing up here. So we will have one more enzyme involved. This enzyme's name is DNA ligase. And what DNA ligase does is it joins these fragments together. So DNA ligase makes that last phosphodiester bond, which is going to then give us a nice solid continuous piece of DNA. And notice that this process will just continue on and on and on. Now, if we divide this replication bubble in half, notice that on the bottom here, this strand, we started with one primer and then we're off. Nice long continuous piece of DNA. This is called the leading strand for DNA synthesis. It is leading because the 3' end of new DNA proceeds towards the fork. If we look at the top one, this top strand is going to be what we call the lagging strand. This is the lagging strand because we have just the opposite thing happening. The 5' of the new DNA faces the fork. And keep in mind, we can't add on to the 5' end. So because we cannot add on to the 5' end, we have to keep adding primers there when we're talking about the lagging strand. This means that the lagging strand is going to be synthesized in a lot of little pieces. It's going to require the involvement of a lot more primase, a lot more ligase as well, and it's going to take longer. Now, if we look at the other half of the replication bubble, notice that everything is flipped. This is due to that anti-parallel nature of the DNA. So over on this side, the top would be leading and the bottom would be lagging. We want to be able to recognize which strand is going to be the leading versus the lagging, top versus bottom. Make sure that we're looking at the new DNA that's being synthesized and which end of that new DNA, 5' or 3' is actually facing or going towards the replication fork. Now, one last enzyme that we have not mentioned is the topoisomerase. Topoisomerase is an isomerase enzyme. Isomerases make isomers. So this one is going to make some isomers, not going to significantly change anything. But what this enzyme does is it's going to circle the DNA back behind what we would call the replication fork. And it does this because as we start to peel apart the two strands of DNA, the DNA becomes a little bit tangled. Because it becomes tangled, the topoisomerase is going to actually cut one of those DNA strands, allow it to untangle, and then it is going to seal it right back together. And we will have the same thing happening over here. It's going to cut the DNA strand, allow it to untangle, and then it is going to join everything right back together again. That's the process of DNA replication. Now, there's a lot of enzymes involved in all of this. I know my picture is not the most beautiful of all pictures, so I would definitely encourage you to look in your textbook at the pictures that are shown to you of the DNA replication process, as well as look at the animations that are posted on Angel with regards to Chapter 16 so that you can see the process in motion and hopefully understand what is happening with the different enzymes involved in DNA replication.